Hi students, this is Mandri Kumar Rajesh. In this video, we are going to discuss the structure of a bacterial cell. Here, I have drawn a typical structure of a bacterial cell which shows different parts. So in this video, I am going to tell about the structure of bacterial cell and its parts one by one. First, I am going to tell about the capsule or glycocalyx. Some bacteria surrounded by gelatinous substance or mucilaginous substance which are made up of polypeptide or polysaccharide or by both, and they are called as glycocalyx, and that layer is called as glycocalyx. A thick layer of glycocalyx bounds to the cell wall, tightly bounds to the cell wall, and that layer is called as capsule. This capsule prevents the bacteria from desiccation. Desiccation means dry condition. So it prevents the bacteria from desiccation and keeps the bacteria moisture, and it prevents the bacteria from antibiotics. So due to the presence of capsule, the antibiotics are not able to affect the bacteria. So capsule prevents the bacteria from antibiotics. Then due to the sticky nature of the capsule, it can attach to the substrate of plant roots, human teeth and tissues. Then it helps in retain the nutrients in the bacterial cell. So the capsules helps in retain the nutrients in the bacterial cell. So that's all about the capsule or glycocalyx. Then I am going to tell about the cell wall of bacteria. The bacterial cell wall contains granular and it shows rigidity. In gram-positive bacteria only, we are able to find granules. While in the case of gram-negative, granules are absent in the bacterial cell wall. So the granules are present in the gram-positive bacteria cell wall and gives rigidity. Then the bacterial cell wall provides shape to the bacteria. So this cell wall provides shape to the bacteria and it gives protection to the bacteria. The cell wall gives protection to the bacteria. Then the chemical composition of the bacterial cell wall is peptidoglycan or mucopeptide. Then one of the most abundant polypeptide which is present in the bacterial cell wall is called as porins. So porins is nothing but a protein which is helpful in the diffusion of solute, which is helpful in the transport of solute. So porins make a pore. Through that pore, diffusion of solute takes place. So porins are very, very important in transportation of solutes. So that's all about the cell wall. Then I am going to tell about the plasma membrane. The bacterial plasma membrane is made up of lipoprotein. So due to the lipoprotein combination, the plasma membrane controls the entry and exit of small molecules and ions. Due to the lipoprotein nature of plasma membrane, it controls the exit and entry of small molecules and ions. And the plasma membrane contains enzymes which are involved in oxidation, metabolism, and which contains enzymes which are involved in photosynthesis. So the plasma membrane which contains enzymes which are involved in oxidation, metabolism, that is respiration, and it contains enzymes which are involved in photosynthesis. So that's all about the plasma membrane. Now I am going to tell about the cytoplasm. So in the bacterial cytoplasms are semi-transparent. They are thick and semi-transparent. It contains inclusions and ribosomes. So the cytoplasm contains the ribosomes and other inclusions. The cytoplasm inclusions like glycogen, then polybeta hydroxybutyrate, then sulfur granules, then gas vesicles. So these are all some of the examples of cytoplasmic inclusions. So glycogen can provide energy when bacteria is in need of energy. So glycogen converted into glucose and it provides energy to the bacteria. Then polybeta hydroxybutyrate also act as a energy provider, when the bacteria is in need. Then sulfur granules means aggregation of microorganism. Sulfur granules means aggregation of microorganism, especially progeny microorganism. Then gas vesicles. So in the phytoplanktons, they contains gas vesicles which are helpful in the buoyancy. So the gas vesicles are present in the phytoplanktons, that is the free floating algae which contains gas vesicles which are helpful in the buoyancy. So that's all about the cytoplasm. Then I am going to tell about the genetic material of the bacteria, that is nuclei. So the genetic material of the bacteria is not well defined because the nucleus is not surrounded by the nuclear membrane and the nucleus is not well defined. Then the bacterial chromosomes are single-stranded circular DNA. The bacterial chromosomes are single-stranded circular DNA which do not possess nuclear membrane and which do not possess the membrane-bound organelles. So that is why the genetic material of the bacteria is said to be nucleoid or genopore. Nucleoid means they are not well-defined nucleus and they do not possess the nuclear membrane. Genopore means it contains the chromosomes and then do not possess the histone proteins. That is why they are called as genopore. Then I am going to tell about the plasmids. So plasmids are otherwise called as extra chromosomal material. The plasmids are otherwise called as extra chromosomal material because they can self-replicate, they can act autonomously, and they are circular in nature. The plasmids are circular in nature. They can resist to antibiotics. And toxic materials. And plasmids can produce bacteriocin. So it is a toxic substance or a protein which inhibit the growth of related bacteria. So bacteriocin is a protein which is produced by the bacteria which controls the growth of the related bacteria. So plasmid can produce bacteriocin. The plasmids can be classified into different types based on their function. If the plasmid which are involving the fertility, that means the plasmids which are involving the transfer of genes, the plasmids are said to be F plasmid. If the plasmid which are involved in gene transfer, the plasmids are said to be F plasmid. Then if the plasmids which are involved in resistant to the antibiotic, if the plasmid shows resistant to antibiotics, that plasmids are said to be R plasmids. Then if the plasmids can produce colicin, so it is a protein which can inhibit the growth of related bacteria. So if a plasmid can able to produce a colicin protein which can inhibit the growth of related bacteria is called as plasmid. It's called as plasmid. Then the plasmid, it can induce the growth of root is called as Ri plasmid. If a plasmid can able to induce the growth of root is called as Ri plasmid. Then the plasmid can cause tumor, can induce tumor, that plasmids are called as Thi plasmid. If a plasmid can induce the tumor in an organism, and that plasmids are called as Thi plasmids. So that's all about the types of plasmids. Now I am going to tell about the mesosomes. Mesosomes are nothing but infolding of plasma membrane, produced in the cell in the form of vesicles, tubules and lamellae. So the mesosomes are nothing but infolding of plasma membrane produced in the cell in the form of vesicles. Vesicles means they can secrete some enzymes. Then tubules and lamellae. Lamellae means that structure which is involved in respiration. So the mesosomes are involved in respiration and binary fission because plasma membrane contains oxidation metabolites. Contains enzymes which are involved in oxidation metabolism. So mesosome contains enzymes which can involve in respiration because in bacteria there is no presence of mitochondria. So due to the absence of mitochondria, cellular respiration takes place in the mesosomes. Cellular respiration takes place in the mesosome because it contains the enzymes which are involved in oxidation metabolism. And mesosomes can involve in binary fission. Mesosome can involve in, helps in the binary fission. Then I am going to tell about the ribosomes, that is polyribosomes. So ribosomes are otherwise called as site of protein synthesis. Ribosomes are otherwise called as site of protein synthesis. The number of ribosomes varies from cell to cell. That means it may ranges from 10,000 to 15,000 ribosomes. So it shows great variation cell to cell. So ribosomes are otherwise called as site of protein synthesis. In bacteria, it shows 70s ribosomes. So it contains two subunits. The larger subunit contains 50s, smaller subunit contains 30s. So S means Svedberg unit. Then the 70s ribosomes is the characteristic features of bacteria or prokaryotic cell. Then these ribosomes are attached or remain held together by mRNA. So this is the larger portion of the ribosome. This is the smaller portion of the ribosome. This is called 50s. This is called 30s. So these two ribosomes are held together by a structure called mRNA. So the two ribosomes are held together by a structure called mRNA and it forms polyribosomes. Or polysomes. Then I am going to tell about the flagella. So flagella are formed by a protein called flagellin. flagella are produced by a protein called flagellin. Then the flagella, the bacterial flagella are cytoplasmic in origin. The bacterial flagella are cytoplasmic in origin. Then the flagella are helpful in locomotion of bacteria. Based on the number and position of flagella, bacteria can be classified into different types. We already discussed the types of bacteria according to their position and number of flagella in the last video. Then I am going to tell about the pillars or fimbriae. Pili or fimbriae are hair-like appendages which are present on the surface of the cell wall of gram-negative bacteria. Example, Enterobacterium. In addition to normal pili, a special type of pili called sex pili, which are involving conjugation, that means which are involving sexual reproduction. So that's all about today's video. If you have any doubt, please clarify with me. Thank you very much for watching this video. Thank you.